Yeah, welcome everyone. I warmly welcome all of you to the Fusion Talk series that is organized by Prescience in Silico Private Limited. The company Prescience in Silico, it was founded by academicians from IIT Kanpur and NCL Pune with a mission to bridge the gap between academic research and industry. We do collaborate with both academic and industry institutions and leaders to tackle challenges in drug and material discovery. And we take an immense pride being one of the first companies in India that to develop an in-house drug discovery platform integrated with advanced AI capabilities. And our goal is to make drug and material discovery more accessible, revolutionizing the scientific landscape and driving groundbreaking innovations. Little about Fusion Talk series. It was came out of our wish to bring together academic and industry experts to share their valuable insights into the rapidly evolving scientific field. Through these discussions, we aim to foster knowledge exchange, and inspire new ideas that pushes the boundaries of research and development. The speaker for today's talk is Dr. Gautam Das, who is a co-founder and chief executive officer of genomics company, which is called miBiome Therapeutic LLP, that specializes in healthcare, pharmaceuticals, agriculture, and biotechnology. Um, okay, he's also a chief scientific advisor of Microbiome Research Private Limited. He has done his master's in animal science from University of Hyderabad, PhD in molecular microbiology from ISC Bangalore, and his postdoc research is in genomics and cancer at University of Massachusetts Medical School, USA. He has over 17 years of post-PhD experience and has multiple peer-reviewed high-impact publications in top-tier journals. He worked at R&D division of Reliance Industries Limited as assistant vice president and head of biomaterials, synthetic biology. He's also serving as a member of the advisory and monitoring board of government appointed committee that has served as scientific consultant for reputed NGO and as a member at RIL, Joint Technical Committee and Institutional Biosafety Committee. Um, and today he is going to speak about his company and his research that he is currently working on. And the title of the talk is "The First, Second, and Third Genomes of Human Health and Disease." And we'll be looking forward to this interesting talk. I urge all the audience to put their questions in the chat box, which will be answered at the end of the talk by Dr. Gautam Das. um, Over to you, Dr. Gautam Das.
Thank you. Thank you, Anil. Thanks, first of all, to Dr. Seema here, who invited me to this Fusion Talk series and introduced Pre-Science in Silico to me. It's a very nice platform working with academics as well as industry. I really like that. That's one of my passions to working with academia and industry. I think that is where groundbreaking discoveries can come when we work together. So thank you, Anil, for a very nice introduction. And I will go straight into my talk, which is the first, second, and third genomes in human health and disease. Now, before going to what this is all about, you guys must be wondering what is all this first, second, and third. So, first genome is your own genome, what you are born with. Second genome is the whole bacteria, fungi, archaea, nematodes, all that live with you. That is the second genome, the microbiome, what we call it, the microbial world that lives with us. And the third genome is something that comes as a surprise to us. For example, COVID-19 came or tuberculosis comes in. These are genomes as we call it now. So I would like to introduce you all to this first, second, and third genomes. And our company, incidentally, works on all three genomes. the human genome, the microbiome, and the pathogenomes in human health and disease. Uh, so I'm sure all of you are introduced to genomics. I mean, these days we don't need introduction to genomics. Just few years back, when I used to go to talks, I think we had to introduce what is genetics to what is genomics. Things have changed. Uh, Today, after COVID-19, I think people know what is genomics, people know what is genetics, and that makes our job easy. But what is important, as we started talking in the beginning, is this is an interface of academia and industry. So what is interesting is that if you look at DNA sequencing cost, or the crux of all genomics, which is sequencing, uh, 22, 23 years back, it was about $1 billion to sequence a human genome. And today, uh, as we are talking, we can sequence this at 100 to $200. So that is the reduction in DNA sequencing that has taken place in less than 25 years. I personally feel I have been part of this journey doing genomics at my postdoctoral research and now, and this along this journey, what we have witnessed is something really historical. That how how could a technology evolve so fast that human genome, which costed $1 billion 20 years back, now costs about $100 to sequence it. That's the power of technology. That's the power of genomics, biology, life sciences, and that's where you guys at Pre-Science in Silico can make a huge difference by deploying technologies of this kind. Now, as we are an industry and we are talking to people, the audience mostly come from industry, academia. The market of genomics is very, very important. The way it is growing now, this is something all of us should discuss, that the global genomics market, which was just $18 billion in 2019, is growing at roughly 20% CAGR. And this is soon going to be $100 billion very soon. Next year, probably the announcement would be $100 billion. So that's the speed at which genomics market is growing. Uh, what is driving all this genomics market? How is it growing so fast? It could be the first reason that I feel is the reducing cost of sequencing service that is happening. Like you just saw $1 billion to sequence a human genome to $100 to sequence the same human genome. Rising government support and initiatives, you read all around in newspapers, social media about NHS funding more and more genomes, Indian government funding the 10,000 genome, Arab governments, US, of course, has been the leader in this. Every government across the world is spending on genomics. And that is why, because genomics is so important to us. And so these are the drivers, and then personalized medicine, and I would come to it, is growing. There's a big demand for personalized medicine across clinical disciplines, and I would give very prime examples of where this is being used today. So, uh, Anil introduced our company a little bit. So we are based out of Mumbai. We are a genomics company. We take pride in calling ourselves deep science and deep tech startup company. We specialize in microbiome research. That's where we started out with, and I'll be talking about that: diagnostics, genome sequencing, genome-based diagnostics, bioinformatics, genomics research, and I'll be talking about a couple of products that we have developed. And we set up a lab in the middle of pandemic. And this I always like to say, because this is one of the toughest things that we have done. Establishing and beginning a startup and in the middle of pandemic was really tough. And I'm very proud here to uh, m- announce to all of you that the company has now generated about 50 trillion bases of genomic data, which is the yardstick of any genomics company, how it is performing. And we have processed over 10,000 genome samples, delivered over 1,000 genome projects, etc., etc. So our machines, for example, our MySeq machine that we initially started out with has completed 200 sequencing runs. Our NextSeq Illumina machine has completed 50 sequencing runs recently in about 6 months of operation. We have a team of now 30 people working in the field of molecular biology, computation biology, NGS, microbiology, software, and business professionals all combined. We have been actively filing patents, publishing papers, etc. So, what I would like to suggest here, I wouldn't go into the details of our team. Me and my co-founder Ravi uh, co-founded this company, miBiome Therapeutics, in 2019, start of 2020. And Nandini heads our lab operations and science team. Sandeep heads our business. And we have an excellent team of scientists, technologists. We have a support team, sales, marketing, and finance are all our important pillars of the company. So as I like to say that it takes a village to raise a startup. What is African proverb that it takes a village to raise a child? I would like to put it across that it really takes a village. It's a lot of hard work. Students out there, startups, aspiring for startups, really a lot of hard work, but really, really rewarding at the end of the day. Uh, at miBiome, as I said, we focus on first genome, second genome, third genome. So now what's this all about? The first genome, as I said, consists of our own genome, so non-communicable diseases like cancer, reproductive health, inherited diseases, rare diseases. We focus on HLA typing, transplant genomics, etc. Into the pathogenomes, we work on infections of bacteria, fungi, virus, COVID-19, we have been working HPV, etc. We work a lot on drug resistance, which is a major problem in India. We work on hospital microbiomes. And in the second genome, which is the microbiome, we work on gut, vaginal, oral, skin, scalp, respiratory, you name it, we work on such microbiomes. So many, many more products, services, platforms come along the way in our company, and all of it we try to use India-specific genome information whenever we get. And these are a couple of our research achievements wherein we have been publishing actively, being a private company in a commercial mode. It's very difficult to keep up with publications, but we have been really, really trying our best. 7 publications so far in different journals, review articles, book chapters, research articles, etc. To all of you who use genomics, these are our services, and happy to talk one-on-one on any requirements that you have in the clinical space, in the research space. And who uses our services? These are doctors, diagnostic labs, hospitals, academic research, pharma companies, major corporates, consumer companies, and the government. Now, coming to the topic of today's talk, the first, second, and third genomes. So now, what is the first genome? As I said, this is our genome. This is the DNA that we are born with. So we at miBiome, we do clinical genomics, and research and translation in the field of clinical genomics is what we do. We provide our clients, partners, it could be any hospital, a diagnostic lab, a research company, or companies like you, uh, with state-of-the-art research solutions for disease diagnosis, secondary findings, and therapeutic recommendations. We handle translation research projects concerning clinical subjects to develop prototypes, actionable insights into various genetic and infectious diseases. And we always use, as I said in the beginning, India-specific databases. These are very different. Our population is very different. Our ethnicity is very different. So population-specific differences are very, very important in clinical genomics. This is briefly the scope of work. The samples are collected, the samples are processed in the lab, data is analyzed, very important component. Samples are reported to clinicians and data is stored. So this is the brief workflow in our clinical genomics portfolio. This is how the DNA sequencing pipeline works. You have DNA extract in the lab. We sequence it using many of our machines. We have all machines that you can talk about: Illumina, MySeq, NextSeq. Now we have NovaSeq, we have Oxford Nanopore, Prometheon, MinION, etc. So now this is all the standard bioinformatics that goes into place. We have a team of very qualified bioinformaticians. Now, recently you would have seen that India has published the 10,000 whole genome samples recently. Prime Minister has announced this. Now, a question that arises, is India decoded in terms of genetics? And my answer, if you ask me, is no, because 10,000 genomes for a country of 1.4 billion people is minuscule. We need more and more genomes to even call average. And I'll come to that. Why should we call average? human genome. So we have more work to do, and students here, we really have more work to do. Some case studies from miBiome, and I wouldn't go into specific details, but just for all of you to know that there's some very interesting case studies that come along, and I would like to present a couple of them. Uh, these are some case studies with the exome, and then I'll come to the microbiome case studies that are we do here. So this is one case study wherein we really went as a company very deep into, we convinced the doctor to really go deep into the patient's sample. It was a case of colorectal cancer, and we were able to do multi-omics here. We were able to do genomics, transcriptomics, microbiome. And within genomics, we were able to do whole exome sequencing, 20 gene panel, 52 gene panel, 500 gene panel, both DNA, RNA. Transcriptome, we were able to do 16S metagenomics, we were able to do stool microbiome, tissue microbiome, the microbiome that resides within the tumor tissue, uh, liquid biopsy, that is the cell-free DNA, everything. So from a single patient, we were able to derive data from stool microbiome, whole exome, cancer genes, whole transcriptome, and the liquid biopsy. And this was a very classic case wherein we were able to show that how microbiome dysbiosis is related to colorectal cancer, number one, how mutations in P53 and EGFR do contribute to colorectal cancer progression. And then we were not just seeing this in the tissue, we were seeing this in the liquid biopsy samples too. So pathways that are upregulated, downregulated, these are details, but yes, what we can derive, the information that we can derive, what I wanted to put the point across is the point, information that we can derive from a single patient sample in understanding genomics, transcriptomics, microbiome, and much, much more. And today's technology allows us to do this in great detail. Another case study wherein a case of hereditary hearing loss came and exome sequencing was done. First, the child's exome sequencing was done, and the parents' exome sequencing was done, and we were able to conclusively say that this mutation in SLC, which was not earlier reported in many, many patients, we were able to clearly say because this family had that mutation, and conclusively, our genetic counselors were able to say that this was a case of solute carrier mutation in the hereditary hearing loss. Another case came of late onset Bartter syndrome type 2, and the patient had severe hypocalcemia. This is an autosomal recessive disease. It has potassium problems, salt wasting, alkalosis, calcium problems, etc. So now the symptoms, the patient had muscle weakness, fatigue, etc. So we took all this data and of hypocalcemia, hypokalemia, hypercalciuria, and we were suspecting CASR gene mutations, which are classic of this disease. And this is what the doctor told us. But then we did whole exome sequencing in an unbiased way, as we should do as scientists. This had something different to offer. The whole exome sequencing of the trio, that is the child and both parents, showed that there was a mutation in KCNJ gene and clearly showing mother, patient, and father. And what it came down to is there was a compound heterozygosity in this case. And these are live examples, real life scenarios wherein you could pinpoint the mutations to parents and the child, wherein compound heterozygosity was involved. So different mutations in both the parents leading to protein dysfunction of a single protein. That was happening here. Uh, there was literature on this particular gene, but not exactly the mutations that we found. So we deposited those mutations into databases. This will help a lot of clinicians across the world to use this as genotype phenotype uh, correlations. So these were some of the articles. And then what we did in our company is something to do with the second genome. Now that is very interesting. These days, a lot is being talked about microbiome. So now, what is the microbiome? As I like to call it, it is the second genome, and this is where all the bacteria, fungi, archaea, nematodes, protozoa, anything you see the microbial world inside our bodies, that's the microbiome. Now, for this audience, before going into the subject, in my free time, I like to write poetry. And this poetry, particularly this poem, will explain what the microbial world inside us means. The poem is very small, it's a 30-second poem. It goes like this: Tiny trillions inside us. Who is powerful, they or us? We humans think we got guts. The invisibles think they are the guts. We believe our master is brain, but gut is called the second brain. We are all born with our genome, but did you know the second genome? Time to pause and ponder today while we celebrate World Microbiome Day, when mighty trillions shape us better, 3 billion bases perform better. So, this is where what I would like to emphasize is this tiny trillions, the microbes, the microbial world within us. Uh, For example, many tissues have microbes, but why gut is being talked about a lot is this is the epicenter of everything, health and disease. When I call the second genome of health and disease, this is 95, 96% of all of it. Gut microbiome is made of trillions of bacteria, virus, fungi, archaea, protozoa, as I told you. To understand what makes a good gut, and researchers like us all around the world are looking to investigate the gut microbiome in the best possible way. And as I introduced in the beginning, the average is very, very important before jumping into any conclusions. Like in our population, what is the average gut microbiome looks like? And we in our company have been focusing very, very hard to working really hard to actually develop an average gut microbiome. And that is very difficult because the data, not much data doesn't exist in India. So genetics, lifestyle, everything complicates: diet, geographical location, age. So now if you look at gut microbiome, what does it do? As I said, it affects everything: liver disease, IBD, inflammatory bowel disease, obesity, diabetes, cancer, infectious diseases, even diseases like psychological diseases, mental health. There's a gut brain axis, autoimmune diseases. Everything can be now correlated very well, very scientifically to gut microbiome. This is an example, another case study wherein you see normal control having firmicutes, bacteroids, etc. Disease cases: type 2 diabetes, inflammatory bowel disease, obesity, metabolic syndrome. Now we have sequenced about 5,000 microbiomes in our company, and we have numerous cases of dysbiosis like this in disease. So what is the solution? Now I have stated the problem of microbiome, what it does. What is the solution? In our company, we have developed a product, and this was launched in 2022 after 2 years of a lot of hard work forming the baseline that I talked about. And we came up with a product called My Gut Quotient, or My GQ, which is, we like to call it, it's a journey to a healthy, happy gut. So now you have all these studies, genetic and functional characterization of microbiomes to genomes, in vitro, ex vivo studies, gut microbiome data, model studies from rat, mouse, etc., clinical studies from humans, the studies on probiotics, chronic disease management, research on microbiome, restoration of gut, everything combined into an central engine to give you personalized recommendations for diet, that is food, prebiotics, probiotics, and to give you a overall gut health index, what we call the average baseline. So this is what we did, and we developed this product called MyGQ. As I said, the nice journey to a healthy, happy gut. I would encourage all of you to just log in into our website. We have a special website for this, microbiome.in. Very easy to remember. We are in India, so microbiome.in. This is a journey to a healthy, happy gut made easier through a single click. You go there, you select the test. There'll be a lot of questions, which test is better for you. And then the kit comes to your home. You give the stool sample. The kit comes to our lab. We do metagenomics, shotgun metagenomics, 16S metagenomics, and then we analyze the data. We recommend food, prebiotics, probiotics, and where are you falling when compared to the average Indian? And how did we do it? By sequencing lots and lots of Indian microbiomes. So that is what we have done in our company. And how do we do it? how do we sequence the second genome or microbiome, the analysis workflow goes like this, and I'm happy to discuss more with any of you present in the audience. The pipeline goes like this. We extract DNA, we sequence it. There are markers when we use shotgun sequencing. There is 16 is amplicon that we use when we do amplicon sequencing. A lot of bioinformatic tools are used to determine microbial abundance, microbial function. A lot of statistics goes into this to determine the microbial diversity, abundance, etc. So on the right is an overview of statistical and visualization methods. You don't need to go into the details, but what is important is that what I wanted to say, and especially for this audience, that a very thorough and rigorous analysis is required. A lot of statistics is required finally to arrive at conclusions from a single stool sample, whether your microbial abundance is dysbiosed or very good, you have a good microbiome, the best microbiome when compared to the average Indian. So this is what we do in our company about the second genome, the analysis workflow that we use. Microbiome, as I said, is everywhere. It does not stop at the gut that I just now described. Uh, Dysbiosis of microbiomes can cause oral diseases of the dental cavities, etc. Respiratory diseases, like even asthma, has been correlated now to respiratory microbiome dysbiosis. Vaginal microbiome is very, very important, and we have a very strong focus on vaginal microbiome in our company. Uh, Skin microbiome, many of the dermatological diseases, and gut microbiome I already talked about. So as you can see, it's everywhere, right from starting from the oral to respiratory, vaginal, skin, gut, everywhere. You have lung microbiome, urinogenital microbiome, etc. So you name it, you have it, and the microbiome is actually doing everything inside our body, outside our body, like in places like skin too. In our company, we focus on many R&D projects, and it is very difficult to invest in R&D, as you all will appreciate, but we try to do our best to do R&D into many areas. Like obesity is one of the areas that we did a R&D project on. 24 subjects, obese people were taken. IBD, we have a bigger number, 120 subjects were taken, and inflammatory bowel disease and gut microbiome, one of the first studies from India on a commercial level is happening here. Dr. Rahul Shah is our collaborator. Vaginal microbiome with Dr. Deepak Modi at National Institute of Reproductive Health here. We have been collaborating on 75 subjects for preterm and full-term births. Vaginal microbiome is so important that it can lead to preterm births. Respiratory microbiome, as I said, we did a work on 50 patients here. And lastly, we talked about the first genome, that is our genome, the DNA that we are born with. We talked about the second genome, that is the microbiome or the microbial world that resides within us. And lastly, the invader genomes, the pathogens that invade us every day. Now, in our company, we have, in India particularly, we have lots and lots of infectious diseases to deal with. In our company, we chose a couple of diseases. We cannot be working on everything. So we chose a couple of diseases wherein we were very clear that precision medicine is possible. And one of them, as you all appreciate, is tuberculosis. As it is a global threat, 10 million people fall ill even today with tuberculosis. The numbers are alarming. 1.5 million die from this disease even now, and that's about 10 to 15% death rate, and that's very high for any disease. Uh, Worldwide, this is the 13th leading cause of death, second leading cause of infectious diseases after COVID-19. This is way above HIV and AIDS. So India leads this disease. So that is a wrong place to lead, but India really leads this. And the 30 high TB burden countries account for about 86% of all TB cases. Now that's sad news. The WHO statistics gives an estimate of about 2.5 million cases in India, and out of which again 10% would die. So imagine even today, 2.5 lakh people die with tuberculosis per year. multi-drug resistant TB. If TB was a strong disease, now we have another stronger problem called multi-drug resistant TB. Now, this remains a public health crisis, and this is a big health security threat. And in many forums, I call this as the next pandemic. And why do we call it as this pandemic? It is a silent pandemic. Multi-drug resistant TB is everywhere. And what would happen? I wouldn't want to scare you, but really, this is something all of us should think about to solve this problem. So we took a stand in the company that we will work hard on tuberculosis, as quick decision making for the right drugs is the key in the treatment of this disease. So we developed a panel, a targeted panel. There were many solutions available in the market. On the left side of the slide, you can see all the solutions that were available, but cost was an issue. So we had to develop a cheaper, faster, and better method. When I say cheaper, it is probably one of the cheapest solutions for 15 drugs available in the market, and faster because it works directly from the sputum. No culturing is required. So a test that would take 42 days or even 5 to 6 weeks to come, report to come, can be done in less than 3 days now. That's the power of NGS. That's the power of genomics for students. If anyone is there, look at this power of genomics, how it is bringing the time from 6 weeks to 3 days. And it is better because the depth of sequencing can be very high. It's a small panel, just 15 drugs, 10 kb panel, and these are multiplexing PCR followed by sequencing. So we give doctors a turnaround time of about 2 weeks because you need samples to pool here. And from our lab, actually, this can be done in less than 3 days. So this is a proof of concept study that we did with a leading lab here in Mumbai, wherein blinded samples were taken, DNA was extracted. The whole sputum DNA extraction we had to optimize because there was no available sputum DNA extraction for targeted NGS available. We developed the whole multiplex PCR assay. It looks simple now. There's a lot of work gone to develop it bioinformatically first, and then to do several iterations of this panel inside the lab. So there's bioinformatics suggesting something, we're trying in the wet lab, and the iterations go on. We do NGS sequencing, and finally the reporting. The software was also made at miBiome, wherein data analysis can be given in seconds. So this is what we have been doing in the infectious disease area. Another important disease uh, caused by human papilloma virus. As you all will appreciate, this is a very, very important virus to talk about because cervical cancer, the disease that human papilloma virus causes, is the second most common cancer in Indian women. 6.6% of our population suffers from one or the other sexually transmitted infection. Now, this is a big number. Almost 7% of people are suffering from sexually transmitted infection. And one of the leading viruses in this is human papilloma virus. There are several risk factors how you can get this virus. There are many, many symptoms. I wouldn't go into the details. There is testing available, RT-PCR based, PCR based, pap smears, as women do regularly. HPV test based on PCR is available. But then every test has its own weaknesses. For example, the breast PCR test also takes into consideration only a couple of HPVs, the high risk HPVs, and some of the low risk HPVs. So we thought, let's use NGS to its best. And when I say NGS to its best, it is to sequence everything. And there comes a solution wherein what we do here is to detect all HPV types, and these are 28 types and 13 sexually transmitted infections, all in a single panel. Now, this is what at miBiome we have started sequencing of 28 human papilloma viruses, 13 sexually transmitted infections, all in one panel. Give the doctor a complete picture, a comprehensive picture of what is happening in the sexually transmitted infection. To all students, and everyone in the audience, actually. Today, we hear about AI, ML. Uh, uh, we hear every day about OpenAI, DeepSeek, and everything. And I would like to emphasize that genomics is one of the very nice areas wherein a lot of AI ML can be used. And to do that, the first step would be bioinformatics, that is to analyze the genomic data to understand what is going on. So when technology meets science, science meets technology, I say that magic happens, wherein the wet lab work or the genome sequence is taken and analyzed into today's using today's best tools of computation. And we use this in our company. We use the best tools for DNA sequence analysis, variant calling, diseases like cancer. We do tumor mutation burden, structural variants, gene fusions, small nucleotide variations, copy number variations, indels, and which is insertions, deletions, fusions, etc. So all of this can be nicely analyzed today because the human genome is available. The 3 GB human genome that was decoded is available, and you can do lots and lots of things. Many pathogen genomes are available. The entire microbiome sequence is available. Average Indian microbiome is available through us. So you can do lots and lots of things using bioinformatics, a very important subject to study in the life sciences in today's age. And coming to what I just discussed, AI ML is exploding so well that we all have to use it in the best possible way. And as Clive Humby, a famous mathematician, described, data is the new oil. And what better can it be than genomic data? I mean, we talk about e-commerce data, we talk about retail, traffic data, etc. But what better can it be to take this genomic data and use it to healthcare? Because the magnitude of data that is arising in the public, private, and commercial databases is unprecedented today. Look at healthcare data. I mean, the metadata that comes from a single patient is unprecedented. And you take all this raw data, the genomic data, you clean that genomic data, and you find patterns. You develop models, you train those models, you evaluate them, and you come up with artificial intelligence that never existed to find patterns in the big genomic data sets. To give you a perspective, human genome is 3 GB in size. 3 billion bases is our genome. When we sequence it 30 times to be more accurate. So 90 billion bases are sequenced in a single human genome. So that's the level of big data that I would like to call it. And you take this big data and you analyze using AI ML, and surprising things can come. And you guys at pre-science, I know, are doing a lot. I would encourage you to do more and more genomic data that comes with every patient, and it is related to every disease that we can talk about. At miBiome, we have been doing lots and lots of platforms, products, services, all of it. We are trying our best to do very good research, state of the art research, as we have the state of the art infrastructure like all Illumina machines, all Oxford Nanopore machines. So we try to do services with our partners, academic labs, clients like doctors, hospitals, diagnostic labs, pharma companies, biotech companies, consumer companies. And I'm very happy to share with all of you that we have undertaken almost 1,000 different projects from academic labs and hospitals. So far, we have touched more than 1,000 patients' lives using our clinical genomics portfolio. We have developed a product on tuberculosis, a product on microbiome that I started out with. And we have been developing products on hypercholesteremia, exome-based HLA typing, transplant success, AIML in genomic medicine, and that is something becoming very close to my heart because I really believe that this is the big data that we all should be working on. Cancer, autism, many rare diseases. These are many of our offerings. And on the left side is all our services. Happy to talk in detail with all of you. And going further, we would like to work more on reproductive health, animal biotechnology, plant biotechnology, pharmacogenomics, and many areas that we haven't started working. So happy to talk, discuss anything that you have. Please reach out to us at hello@mybiome.com. Thank you very much.
Yeah,
Happy to
thank
take
you.
questions.
Sure, yeah, thank you, Dr. Das, for very interesting talk. Yeah, it was really fascinating. So we have questions from our audience. I'll take them one by one. The first question comes from Dr. Sanjay. He says, Have you come across any neurogenesis inducing microbiota classes or their secondary metabolites in any of the cases you come across? Also, would like to know about your take on neurogenesis and microbiota.
We have not done so much of the urinogenital microbiome. We have done for our clients. So that is the research project. We do not currently have a product on urinogenital microbiota. There's a lot being talked about the urine microbiome, which was very surprising. I mean, urine was supposed to be free of all microbes, but metagenomics is giving surprises. So definitely there is a microbiome associated with the entire urinary tract, etc. The next question was metabolites. So that is something very interesting. And in today's age, I think the next step of our company also, we would like to take it to a multi-omics approach, wherein metabolites would be studied simultaneously along with the microbiome. Today, we know just this is just the snapshot of what is happening. The microbes, the list of microbes, the genes, or to an extent metatranscriptomics when we call the expression profiles of the genes. But the next step is what are all this doing? What kinds of metabolites are being secreted, and the whole metabolome is the next step. So combining microbiome to the metabolome of the microbiome is the next step forward.
Okay, thank you. The next question is from Akhilesh. He asked, what type of machine is used for whole exome sequencing and how much accurate the results are?
Yeah, very good question. So for people who don't do so much genomics, we have different platforms. We have all kinds of platforms. The highest accuracy platform which is available in the market is Illumina, 99.8% accuracy. We have that platform. We use NovaSeq 6000 for doing exome sequencing, whole exome sequencing. And other platforms like the long read sequencing, for example, when we use Oxford Nanopore, even that has come to about 95% accuracy today. So very good accuracy with both long read and short read. And we, for all variant calling, we use short read, which is 99.8% accuracy.
I see. Yeah, just in continuation to this question, I have one question. So let's say from genome to its sequencing and annotation, how much the time it is required for you guys?
Yeah. Today, Today, the entire workflow is very, very streamlined. We have
Mm-hmm.
automation robots that do it. Uh, Library preparation time is something that is rate limiting. Still that thing takes about a whole working day. So I would say one day to do the entire library prep, and the
Mm-hmm.
sequencing takes 48 hours. So the
Mm-hmm.
whole sequencing of human genome takes 48 hours. It's any
48.
sequencing technology will be 3 days, three
Mm-hmm.
full days of uh, lab work, and the analysis fortunately is very fast in today using high-end computing. The analysis, the variant calling exomes can be done in a couple of minutes.
I see. Yeah. Okay, thank you. The next question comes from Potosima. She asked, it is with respect to the scalability of sequencing techniques at mass level, considering the quantum of data, generated analysis time, cost, how soon do you see this is getting, wider accessibility for majority of populations? And we are talking about skilled workforce to deliver these results. Do you see this as a bottleneck?
Yeah, I will answer the second question first. Skilled workforce is a bottleneck, I would say, and that's a big bottleneck. These are technologies that needs to be done by very skilled workforce. Very high quality data can come in only when experimentalists who are very specialized in doing NGS do this work. Bioinformaticians that are very specialized in analyzing NGS data do this work. So we have really worked hard to assemble the team I showed you. We have people from all top institutes, India and globally. So that is the first thing. And when will sequencing become mainstream? I mean, I would want it now. But why is there still question about acceptance? I would say one reason is cost, but fortunately, that is going down. Every year that we come into sequencing, that the costs are going down drastically. So today we started out with microbiome tests for 15,000 rupees. We are selling microbiome tests for as low as 5,000 rupees today. And consumer genomics is sold at 5,000 rupees. Whole genome sequencing, which was earlier 5 lakh rupees, is being sold at less than a lakh rupees. The entire whole genome information of an entire human being is being sold at less than 1 lakh rupees. So that is the power of technology that is happening. Still, these are expensive. These are not RT-PCR in hundreds, hundreds of rupees. Still, that day is far away. But I'm positive that costs are going down.
Okay. Yeah, so there's another question from Seema. She asks, while genomics is often presented as a solution to many diseases, what are the limitations of genomic medicines in terms of actually treating complex diseases like cancer, Alzheimer's, or autoimmune disorders?
Yeah, very good question, Seema, again. So I think one of the biggest beneficiaries of genomics has been oncology, and there's a direct application of oncology. Any targeted drug you take, you need a sequence before even administering the drug. So that is the level of advancement in genomic sequencing and genomics in particular that has gone, and it has become a companion diagnostic, is the right word, for the targeted drugs in oncology. For neurological disorders, we are not there yet. We know that there could be predisposition of certain genes to certain neurological disorders. Now, what do we do with that? Predisposition is a challenge, and I face that challenge every day as a co-founder of a genomics company. That if I have predisposition, what do I do? Do I take medicines? Do I take care? Uh, I would say preventive health is a coming up as a big concept now, and knowing predisposition is important because you can take more measures, preventive steps to take care of your health. So that is where it would help. Autoimmune diseases, not much. The diseases and autoimmune. Sorry, Anil, I think I lost.
Yeah, I'm back. Actually, my network was a little unstable.
Yeah,
Can you hear me? Yeah, okay. So,
yeah, now I'm able to hear you.
yeah, okay, okay, great. Okay. There is another question from Tushar Kagira. He says, yeah, so he wants a comparison between genome sequencing in India and other countries.
Yeah, you would be very happy to know that the costs are currently almost the same. We have reached a stage wherein India can compete with the entire global community in terms of costs. There were days like only 2, 3 years back, there were days where people used to send samples abroad because the costs were low. Expertise was not there. Expertise is there. We have the costs going down. We can compete any with any company globally, even China.
Okay. Okay. Again, there's one more question from Seema. How about infectious disease, third genome?
Yeah. So drug resistance, for example, it is being already used in infectious diseases, and this is a clinical prescription for NGS that is happening today as we talk. So there is widespread acceptance of NGS-based precision medicine, even in infectious diseases like tuberculosis is something that we work on. Uh, Another topic that I talked about, HPV, we still need some licenses from the Gynecological Society of India to make it as a mainstream test. It is still RUO, research use only. Uh, Other diseases in the research area or surveillance, for example, COVID-19, when it came, the whole variants of COVID-19 came through sequencing. And even today, wastewater is being sampled routinely by the government to find out variants of COVID. Even today, there are variants of COVID coming. So that is the power of genomics in infectious disease, surveillance, management, treatment, and precision medicine like drug resistance mapping.
Okay, thank you. Yeah, if there are no more questions, I'll ask one question from my side. That
Sure.
is, with the increased use of antibiotics, how does it affect the microbiome, the second genome?
Yeah, very good question again. So definitely antibiotics are not good for our gut, and all of
Yeah.
us would have experienced in our own lives that you take antibiotics and the next day you feel sick with your stomach.
Yes.
And that is clearly, we see clearly that it damages your microbiome. Uh, Today, doctors are very vigilant enough to give antibiotics and couple of probiotics along with that, so that your gut doesn't get damaged entirely. So a couple of very good probiotics like Lactobacillus and Saccharomyces boulardii are given along with the antibiotics. But we tell our clients who suffer from gastrointestinal disorders to actually map your microbiome before and after antibiotic. If there is a severe gastroenterological issue, we ask the doctor and the patient to map your microbiome, and we see those results. There is a strong dysbiosis of gut microbiome after administration of antibiotics.
Yeah.
And these are
Okay.
antibiotics not being targeted for the gut to start with. This could be a general amoxicillin kind of antibiotic.
Correct. Yeah. Okay. Okay. Yeah. So there is a question from Dr. Subrata Dev. He asked, okay, so he wants your opinion on human behavior related to gut microbiome.
Yeah, again, very good question. We are working with a couple of leading psychiatrists and psychologists and scientists working in this space of uh, uh, the whole psychology and psychiatry, I would say. Uh, Initially, these things were thought of as a myth, how microbiome, something in the gut,
Right.
is affecting your mood. But if you look at it, 70-80% of serotonin is made inside the gut by our gut bacteria. So, depression or mood or psychological illnesses are connected with our gut because of metabolites like this. Now, the gut brain axis is actually a result of the interplay between the metabolites that come out of the gut microbiome and how do they affect the neurological parameters, the neurons. So that is the interplay that affects your psychological illnesses or even simple problems like a mood disorder or anxiety or depression. Everything. I mean, there's clear scientific evidence to show that upon restoration of the microbiome, people have shown that scientists have shown that these symptoms can go away.
Okay. Okay, so we are running on time,
Very nice
and
question.
yeah, yeah, I see no questions. So yeah, um, I would like to thank Dr. Gautam Das for the wonderful talk, and I'm sure that our audience they have thoroughly enjoyed the talk as well as the discussion thereafter. Yeah, with this, yeah, I would like to ask Dr. Seema to come and give you a word of thanks, and after that, we'll wrap up the session.
I
Seema.
have one last question before I,
Oh,
deliver
sorry.
the word of thanks, Gautam.
Yes,
So
yes, Seema.
I'm just curious that, for the gut bacteria, is there any documented case where the gut bacteria have seen to evolve under, let's say, antibiotic pressure or resistance? and they change into something different, or good bacteria always good bacteria? They just die and don't mutate. How does it work?
Yeah, very good question. And there are only very few studies and something to really examine. And I think our studies will be something which can play a big role. We haven't looked at it that systematically, but we do have data. And thanks, Seema. I would really go into the depth of this because we have shotgun data uh, before and after antibiotics. So we can look into the mutatability of this bacteria. What what you are suggesting is that can a bacteria upon antibiotic treatment evolve in such a way to survive? Very good question. I
There's a
really,
whole
honestly,
there's this whole
yeah,
industry of
this is a very interesting
probiotics
area.
and
Very interesting area.
nutraceuticals, right? I mean,
Correct. I mean,
this could determine how that industry moves forward.
correct. And there is a concept coming up of the gut AMR reservoir. And that came up
Right.
from the suggestion that you gave that if you administer
Right.
antibiotics, you have resistant bacteria to those antibiotics coming up, and that is the reservoir. But
Right.
we haven't looked into that. But we have rich source of 5,000 samples data, shotgun data, that we can look into the mutability of what is happening. Thank you.
Super. All right.
Yeah. Sorry. I'll go with one question. Sorry.
Sure.
Yeah.
Sure,
So
sure.
how does the newborn babies, they get their gut microbiomes? So I mean
Yeah.
they
So the
yeah, please. Sorry.
So there are lots of studies now on the infant microbiome or the newborn microbiome, and we have seen a lot of cases of newborn microbiome, and then there is even complexity. How was the newborn coming? Like cesarean deliveries from normal deliveries, there is a different microbiome coming in. Mother's development, how the pregnancy took place, a different microbiome coming in. And these are interesting. Like one would not expect that a gut microbiome would change, but it is changing. It is there is clear evidence that cesarean versus normal delivery, the gut microbiome is changing, not the vaginal microbiome, not the skin microbiome. So we have to trust the data. But not much solutions have been provided wherein people born with dysbiosed microbiome, if you restore at newborn, I mean, those are difficult things to do, like fecal microbiota transplant, for example, is coming in a big way. And for example, a lot of neurological specialists are working on these solutions to reverse autism, to reverse a lot of neuro degenerative disorders. But uh, newborn microbiome is definitely different. And within newborn, again, cesarean to normal is very different. Very interesting.
Yeah. Thank you. Over to you, Seema.
All right. So I had a gut feeling, pun intended,
Yeah.
that this is going to be a good talk. So thank you, Gautam, for accepting our invitation and delivering such a fantastic talk today. And as a longtime friend, I couldn't be prouder. You've successfully created a platform for innovation in India that's truly making a difference. And especially, with the current relevance that you also alluded to, that it holds under the India Genome Project. It's a beginning. 10,000 is not enough, but this is a first step forward, and then we have miles to go, of course. But thank you once again for sharing your
Yeah.
knowledge, your journey, your vision with all of us. I've no doubt that your story and this incredible work that you're doing will continue to grow and inspire many. And we wish you continued success. Thank you so much.
Yeah, and by
Thank
the
you.
way, thanks for
Thank
the
you, Seema.
wonderful
Thank you,
demo.
Anil.
And yeah, thanks.
Oh, thank you. And thanks to everybody who has attended. Thank you so much.
Okay. Yeah. Bye bye.